is macroevolution and life history. And we are going to get started by hearing about macroevolution in mammalian morphology. All right, Catherine, take it away.
Amazing. Thank you. Hopefully you can all see a nice little meerkat right now. I see a thumbs up. Perfect. So my name is Catherine Sheard. I'm the UK equivalent of an assistant professor at the University of Aberdeen in Northern Scotland, and I'm going to be talking a little bit about mammals today. Maybe, if the slide advances. There we go. Okay, so one of the many really interesting things about mammals is that they span a huge range of size classes, right? You have the Etruscan shrew, which can fit on your palm, clocks in under 2 grams, all the way up to the Atlantic blue whale, which is possibly the largest animal ever to exist on Earth. And a tremendous amount of ink has been spilled about why have mammals expanded so much in terms of body size and what determines why one species may be in one place and not the other along the spectrum, and what are the evolutionary and ecological consequences about being one body size or another. And that is all fantastic. But there's so much more to mammal morphological diversity than just body size, right? Morphology is not a single dimension. There's so much more that can be said about how mammals look different, and especially how their size and shape may vary across the 6,000 or so species that are alive and kicking today. One of the problems in studying this morphology, though, is the lack of data. We want to describe this cute little red panda right here and all its nearest relatives. It's fairly easy to do so in terms of body size, and it's not so straightforward to do this in terms of other morphological measurements. Those measurements exist because people like mammals. They're cute and they're cuddly and so forth, but they haven't often been collated into one easily accessible place. However, a few years ago, there was a global pandemic and I got stuck in a lockdown. So I decided to see how many of these traits I could stick into an Excel spreadsheet while waiting for the world to open back up. So for the purposes of this talk, I'm going to be thinking about what ultimately boils down to 14 traits. The first is body size itself, a minimum and a maximum, right? Both a toy poodle and a Great Dane are dogs, both very valid dogs, but they have both very different body sizes. The next two sets of traits have to do with body length and body height. You can be a certain size, but do you get to that by that size being long and skinny, or do you get to that size by being tall? And again, with a minimum and a maximum for each. And for the purposes of this talk, I'm then looking at 3 additional traits that have to do with extremity length. So ears, tail, and feet, and you'll see why I'm looking at those in particular in a sec, but people like recording them for a whole bunch of reasons, including that they vary a lot across the mammalian class. And then finally, in terms of bats, it's very hard to get comparable traits at a large scale for bats, except for forearm length. So minimum and maximum forearm length as some sort of proxy of shape. Sure, why not? And then armed with these traits, we can think about why these dimensions of mammals, why this way of quantifying our red panda buddy right here might vary across, in this case, about 5,900 species of mammals, though not pulling the wool over your eyes here. Well, I am a little bit, not complete data sets for all species. And if we want to think about how these axes may vary, we can go back in time to the 1840s and think about an idea that was popularized by Carl Bergman, though he was not the first to say it, that these measures of size probably have something to do with temperature. It's the idea that animals will be smaller when it's hot and they'll be bigger when it's cold, right? Because if it's very hot, you want high surface area, low volume to dissipate the heat as fast as possible. And if it's cold, you want to be insulated really well, very low surface area, very high volume. Now, is this at the population level? Is this at the species level? It's a little unclear. People like me start hiding behind a textbook at that point. But this is sort of the general sense of how the relationship between size and temperature should work. We then fast forward to the 1870s, and we enter another sort of fundamental rule, this one attributed to Joel Allen, which has to do with those extremities that I mentioned. So these are, again, things like ears, tail, and feet. The idea being that extremities will be bigger in hot places because extremities are very, very good at dissipating heat, and they'll be smaller in cold places when you want to insulate yourself and keep all that heat to yourself as much as possible. Now, some of you totally on board with what I just said, and some of you are like, wait, what just happened? So to recap, if it's cold, the idea is you want a big body, small extremities. And if it's warm, you want a small body and big extremities. And this has been tested at all sorts of scales and all sorts of levels, but is this true across all mammals with these various dimensions that I showed on the red panda? And there are many complications, one of which has to do with the fact that mammals use their body for other things besides just thermoregulation. And a major one has to do with locomotion. So this is especially obvious for arboreal species. So species that live in trees will often have very long limbs and very long tails, regardless of what temperature they live in, because that's very helpful for like catching yourself on branches, very helpful for balance. And on the other hand, if you're fossorial, so if you dig a lot and you burrow, you're probably going to have especially very small or non-existent ears and tails because that just kind of gets in the way. So I wanted to include locomotion as well. So I also collected data on that. And in particular, I was interested in quantifying the variability in locomotion within a species. So like our tiger friend there, most people would say tigers are terrestrial, and they absolutely are, but like they also climb trees and they also are capable of swimming. And we have methods these days that can take into account the flexibility within a species and traits. So I figured I'd just collect a new data set again. There was a national lockdown. I had some time on my hands. So armed with these traits, approximating size and shape at this level, armed with a whole bunch of different measures of temperature, also with this locomotion, stuck all these measures into various phylogenetic generalized linear mixed models, and then decided to test these ideas from 19th century Victorian white men about how temperature and morphology should relate across all mammals. So going back to our nice little cheat sheet, it turns out that these ideas from the 1800s are absolutely true. On average, mammal species are larger when it's cold and, larger when it's cold and smaller when it's warm. And they have smaller extremities, so smaller ears, smaller feet, smaller tails when it's cold, and bigger ears, bigger tail, bigger feet when it is warm. After controlling for locomotor mode and all sorts of models of phylogenetic autocorrelation. But there's more. This is layers. It's going to be 3 layers of an onion here. There are some exceptions to this, right? Nice little elephant in front of you. To state the obvious, the elephants are big. Elephants are, in fact, maybe bigger than you might expect, given, I don't know, Brownian motion or something, and yet they live in hot places. But as you can see from our elephant friend here, they have very big ears. And this is a really good example of the fact that there seems to be a trade-off between Allen's rule and Bergmann's rule. If you're going to violate one rule, you're actually probably going to follow really closely the other. Above chance and after controlling for locomotion, et cetera, et cetera. So that's layer number 1. Interesting, but probably pretty obvious. Layer number 2 is the fact that I'm just throwing around this word thermoregulation a lot, but there might be 2 different forces driving this relationship with thermoregulation. First has to do with examples like our elephant of big things in the heat. So what do we do to dissipate heat? Are we concerned when it's hot? Or alternatively, are these patterns mostly being driven by small things in the cold? Is this mostly being driven by worries about wintering and insulation and what do we do when it's cold. And I won't go into the details, but it seems to actually mostly be selection for heat dissipation. If you are a big thing, somewhere where it's hot, you're going to follow these rules much more strongly than someone like our lemming here, where like, yes, this pattern is true, but with not as tight of a correlation. That's layer 2 of the onion. And then finally, we have layer 3 of the onion, which is the fact that I've been presenting patterns at the class level. And of course, that's not the only way to divide up biodiversity. And these patterns, Bergman's rule, Allen's rule, the trade-off between them, all of that, do not necessarily hold at the level of major orders. What's going on in the carnivores, primates, bats, rodents, and Artiodactyla, bears very little resemblance to one another and also to the levels across the entire mammalian class. Some of this is to do with the fact that not all of these groups span a very broad range of sizes. Also, not all of these groups span a very broad range of temperatures. But even after we try to filter that out, there's a lot of noise and reversal patterns and just interesting things going on here that kind of defy a simple one-sentence explanation. And interestingly, we also found that when we did a deep dive into the rodents, and specifically rodent tails. We did a different study where we looked at not just rodent tail length, but also their size and their texture and their color and all of that. And we found that patterns that were true at sort of like this population level and genus level and maybe family level were not true across the whole order. What was true the whole order was not true across the major suborders, and that a lot of these macroevolutionary relationships are extremely sensitive to taxonomic scale. So that's what I'm thinking about at the moment. And I really wish I could say banner headline, yes, this is why, but maybe that's what I'll talk about at next year's evolution. So I think I went a little fast, but that's great. More time for questions. If you maybe tuned out or joined a bit late, here's a summary of what I said. Major thank you to my co-author for these global studies, Mike Benton at Bristol. My contact information is there, and I'll gladly take any questions. Thank you.
Excellent, wonderful talk. So at this point, if you have questions, please use the raise hand indicator at the bottom of your screen so that the author can call on you. Because otherwise, I will start asking questions.
Yeah, it looks like there's a question.
Yeah, thank you very much for your speak. But I was wondering if there is any like research about bone density and these type of like trade-offs. For example, I don't know, like animals in hot places are like less dense bones, so they can like store less heat or something like that.
That is a brilliant question. Mike Benton would know my co-author would know the answer to that off the top of his head. I do not. I saw that Joanna Baker asked a question. Maybe she would know this answer. Yeah, I'm sorry. That's great, and I simply do not know. But that's a really good idea. I will see what Joe Baker's question was. What methods did I use? Yeah, so these are mostly run using McMick-Glim so that I could integrate over a distribution of phylogenies. I come from Bird World where that sometimes really matters. In this case, it kind of didn't. In this case, I probably could have saved a lot of time by doing a consensus tree and just running the model once. But live and learn. This is why you test it. But yeah, that's what I did. Oh, yes, using the Upamid trees, yes. Which also cuts the sample size down. I quoted some very large number of measurements we had, and yeah, that wasn't in fact the final number that went into these models. And do the same pattern hold for cetacean? Oh, do I have to stop talking?
No, you have time. You can finish talking, but our next speaker, Albert, you can go ahead and start your screen share to make sure that works while Catherine answers her last question.
Yeah, sure.
The question of do the same patterns hold for cetaceans? I haven't actually explicitly tested that. We did filter out sort of the possibility that purely aquatic species were doing something differently, and that is controlled for within our main models. And I don't remember off the top of my head if that's actually statistically meaningful, but It's a very good question. As you can tell, this is a work in progress. Thank you. I'll stop now.
All right, excellent. So it is 2:45, which means we can start with our next talk. So Albert, feel free to begin whenever you're ready.
All right. Hi, thanks. Hi, everyone. My name is Albert. Currently, I'm a postdoctoral researcher at the Washington University in St. Louis. So over the past year, I've been working on this idea of establishing a general diffusion-based framework for approximating many state-dependent diversification scenarios. So the goal of this research is basically to help us formalizing relationships between evolutionary state patterns, process rates, and mixing type in this model family. So here is the outline of different things that I will go through during these presentations. So let's start with phylogenetic tree in general. So we know that phylogenetic tree shows lineage diversifications through time. So here we can see this is the phylogenetic trees of all bird species through time that is in these publications. And using the phylogenetic tree, we can learn things such as diversification rate. So here we want to learn how this event rate drive lineage diversifications using a diffusion-based framework. So let's talk about the tree generating process. As we all know that when we simulate phylogenetic tree, we start with one lineage sometime in the past, and then as we progress through time, you may have a speciation event where one lineage splits into two different lineages, or you can also have state transition events that occurring along a lineage, or you can also have a species extinction event, and of course things that we observe at the present time. So for this presentation, we'll be looking at the specific class of diversification models where event rates depends on current states. So in particular, we assume that we have the states are discrete. So it's a discrete state dependent diversification model. So what are those models? So in general, this is a joint framework that is used to model trait evolutions and also lineage diversifications in your phylogenetic trees. And as we all probably know, we have this widely used and known BT model, where under this model you have events such as you have energetic state changes and also state-dependent speciation and state-dependent extinction events, which are illustrated on this figure on the top right. And then, of course, you have event rates that are associated with each event under these models. And you also have generalizations where now you can have more than two states. So this is the multi model. Now, these two models, they are part of the special cases of a more general SSE model known as the classy model, where under this model, you basically have all the events that the other two models have, but you also have the phylogenetic state changes, where you also have lineage transitions happen simultaneously as your speciation events. So in this talk, I would like to incorporate a diffusion process to learn about these event rates from an SSE model. So diffusion models is widely used in population genetics to study dynamics of allele changes in the random population. For example, here you have illustrations of populations, non-overlapping populations that are evolved under Wright-Fisher models, and you can ask questions such as, what's the expected time for one of the alleles to fix in the entire populations? But diffusion process is still not widely used to approximate phylogenetic diversification dynamics. It's still quite rare. One example of the publications that use diffusion process for lineage classifications is from the 2016 paper where the author used it to study the dynamics under BC and also MUSI model. So the idea here is that we want to generalize this concept by establishing a general diffusion-based framework to study any discrete state-dependent speciation and extinction model. Okay, so just so why it might be useful. So let's pause for a moment and let's consider like these two biogeographic scenario where you have discrete regions, say region A, and in these regions you have a higher speciation rate, but lower extinction rate. Or you also have another region, region B. In this case, you have a lower speciation rate, but higher extinction rate, and then you may have lower dispersal rate between these two regions. Or you also have other scenario where now your region A has lower speciation rate, but higher extinction, or your region B has higher replication rate but lower extinction rate, and then but you have higher dispersal rate. So which of these two scenarios that corresponds to species richness that you see at the present? Do you see a higher species richness in A than in B explained by one of these two scenarios, or the other way around? So that's the question that we want to answer. So we want to understand the state patterns given model parameters. So the formulations. So here we want to basically simulate forward in time simulation for the number of species across states through times. So what we need first is that given some model parameters from your SSE model, you can derive the diffusion parameters, so just mu I and the sigma I squared for every state in your state space. And then given some initial values, you can simulate the number of species for every state through times under the diffusion-based approach. And you can also do the same for simulating the state frequencies using the diffusion-based approach. Similar, you need to derive the parameters for the diffusion model, and then some starting value for the frequencies you generate forward in time. So here on the left, for example, as an illustration of how it works. On the left, see here we have a stochastic mapping on a phylogenetic trees under an SSE model, where different colors represent different states that a lineage is currently in. So this is the event-based phylogenetic trees. And you basically want to establish these connections with the diffusion-based frequency simulation. Here you have the trajectory for every frequency, for each different state under the model. So what we do is that we basically draw the future value using these equations, the distribution equations, using the current values and the deterministic and the stochastic components of the model. And so to give a context here, we will apply this framework in the parts of a classic model known as the GSE model. So what is the GSE model? So GSE model is basically a diversification model for species range evolutions. So for example, you have this tree here where your data at the present time represents different ranges that each species have. So here you have two different regions, A and B, and species can occupy one of these two regions or both regions. So as usual, we need to also assign events for that corresponds to the model. So under GSE model, you have events such as dispersal, where species can disperse into another region, or you can also have within region speciation, where you have a speciation event in one of the regions. So you can also have extinction events that can occur locally in one of the regions, or it can happen in both regions that leads to a species extinctions. But it can also have a between region speciation, where in this case, at some point in time, you have physical barriers that limits gene flow exchange between two regions. So now start with one species in both regions, you have two completely different species in these two regions. So this is just a little bit of detail about how we simulate the state counts and frequencies under diffusion model. So one thing to note here is that under the diffusion model, as usual, we have assumptions that we start with a relatively large starting condition. So in this case, you can see on the figure on the right-hand side here, we start at this process at this particular time where we have a relatively large plate size, and then we compare the diffusion-based simulation and the tree-based simulation at that point in time. So I'll revisit these figures later on the presentations. And here's just some simulation experiments. So each individual plot represents the diffusion-based and the tree-based simulations for each range state in the three regions GSE system. So you can see that the diffusion-based framework reliably approximates the state-dependent diversification scenarios under this type of model. And this is for the frequencies. So the right-hand side for tree-based and the left-hand side for diffusion-based. We also use some statistical tests to make sure that we are happy with how diffusion approximates the dynamics of states changes over time. So the null hypothesis here is that it's the means of the average of each state count from both approaches are equal, and the alternate hypothesis is that they are different. And you can see that the p-value gives evidence that we cannot reject the null hypothesis. So let's revisit the slide that I had before, where we simulate things at this point in time. So what happens if we simulate under diffusions at this point in time, where you have only one species, which what you will normally do when you simulate a phylogenetic tree? Well, at first the result is not good. You can see that the diffusion-based simulations and then the tree-based simulations do not agree. So what we do is that we have some model-based corrections that will help to correct the diffusion-based paths. So due to the time constraint, I cannot go through these in details, but I'm happy to go through during the Q&A if people are interested. So this is what we have before without applying the corrections, and this is what we have after applying the correction when we simulate things when starting with a single species in random state at your root node. You can see that now we have a very good agreement between the two model, the two processes. The other thing that we can also do with the diffusion-based approach that earlier on I have these two different biogeography scenarios on how species can disperse across regions, and then things that we see at the present time. So your tip state frequencies, so where the ranges for what ranges that each species have at the present times, right? So what we can do is basically we have some theoretical results. I wouldn't go through these in detail, but basically what this result suggests is that we have a way to derive DC rates, given that you know the stationary frequencies of the model, and then vice versa, so you can get the stationary frequencies from the event rates as well. And this is just some immersion experiments, and this one is very interesting here. So the left-hand side here, we simulate things under the two-region GC model, where you have a lower within-region speciation rates and lower dispersal and lower extinctions. And we have another different scenario for the right panel, but they both lead to the same stationary patterns in the long run. So we have an archived version of these research. If you are interested to see more, this one is also has already been accepted in the Bulletin of Mathematical Biology. So what is the future directions for this framework? One thing that we can do here is that we can try to quantify these different evolutionary scenarios for every stationary patterns that you have under an SSD model. So here we have an example of a two-region GST model, where each dot in this simplex represents a set of stationary frequencies, and colors represent the different evolutionary scenarios that correspond to each of these stationary frequencies. And we can do this for any discrete SSE models as well. Lastly, I would like to acknowledge the lab that I'm in, so the Landis lab and all the members. Thank you very much.
Excellent talk. So at this point, we have time for questions. If you would like to be acknowledged by the speaker and able to ask a question, please use the raise hand function at the bottom of your screen and you will be called on. If not, we're going to get me asking questions, which is not necessarily what we all want.
I think there's a...
Perfect. Good, Bjorn. Oh, you are muted. Let me see if I can unmute you.
Hello, can you hear me now?
Yeah.
Yes.
Okay, thank you, Albert, for the very nice talk, and it was very interesting. So I have one question regarding your simulations. So in my work, I've also tried to simulate a bit, simulate phylogenies under the state-dependent birth-death process. And I found that in practice, it can be quite difficult because there's so big variation in the number of like trees that you can possibly get out of this situation. Like any trees between like 3 or 5 taxa, but you can also blow up to something like millions or billions of taxa, given a long time span, even for relatively simple problems. So I was wondering, have you also encountered this issue? And as well, is it also a problem for your diffusion-based approach?
Yeah, actually, this is also one of the main drivers for this research, like you said. So the idea with the diffusion-based simulation is that you try to learn about what state patterns that you try to learn about these state patterns directly from the model parameter. So given some model parameters, can you predict the dynamics of how these states, this state changes over time? So this simulation here is basically just to show whether if you simulate the phylogeny given these rates, would they agree with what we see under the diffusion model, which they are. So that's good. So like the idea is that if you just give me some rates under any discrete state dependent models, we could try to see the patterns of how these state changes over time, and then what's the stationary frequency for these event rates or the other directions as well. Does it answer your questions?
Yes, it does. Thank you very much.
Yeah, thank you.
Excellent. And we have to move on. Thank you, Albert. And thank you, Bjorn, for that question. If anyone has any further questions, I think Albert is planning to stay on in this chat. Otherwise, he is available via the meeting lobby. All right. Our next talk is about to begin on the seed's first theory of the origin of eukaryotic lineages.
Yes. Yes. Good evening. Can you hear me? Okay. My name is Pani Dekosar. I'm a physician, but quite I'm going to bring a new theory of the origin of eukaryotes called the seed's first theory. It's the first theory of evolution since Darwin, and it's part of a more englobing framework called the seed or quantum theory of origins. The outline, we have presented theory, talk about the origins of biodiversity, and we present the evidence, which include the fossil, the viral, and the bacterial evidence, and we'll conclude. The simplest simulation of the seed first theory is that the species of eukaryotes that we know today, sampled here on the top of this chart, they originate from seeds or zygotes that were formed in the greater Precambrian, and those seeds would evolve to different configurations from the seeds to the configuration of the species that we know today. For instance, for the configuration for the lineage on the right side, for the human lineage, we start from the seed, from the zygote to the let's say the common sponge phenotype of early animals. At some later point to Australopithecus afarensis, and later on to Homo erectus, and before which the configurations of Homo sapiens. This is in stark contrast with the theory of natural selection, of the Darwinian theory of natural selection, according to which species, the tapestry of life that we have today, originate from a last universal common ancestor that would diverge, split, and branch out to form the diversity of species that we know today. Where do those seeds come from? Where do those zygotes come from? According to my theory, the first eukaryotic cells were formed by the fusion of bacteria, which was basically gamma proteobacteria, with viruses, which were basically retroviruses, RNA viruses, that would fuse with the bacteria. They would bring the nucleus. They would transduce other genes from other bacteria to the gamma proteobacteria. For instance, transducing the genes from alpha proteobacteria to form the mitochondria in eukaryotic cells. But what about biodiversity? What does explain biodiversity? Where does that come from? We see also that the bacteria involved in the formation of eukaryotes, they are different for animals and for plants. And those are bacteria, they belong to the class of gamma proteobacteria, and they accumulate sulfur for energy, which they oxidize with nitrate. And the bacteria that leads to that gave the animal eggs is Tumagaritha namibiensis. It's a bacterium that was discovered in 1999 off the coast of Namibia by a team from the Max Planck Institute for Microbiology, for Marine Microbiology. They accumulate sulfur, and those white things in the bacteria, they are sulfur globules. And then and actually the name of Tumagaritha namibiensis means sulfur pearl of Namibia. And the bacteria that give rise to algae and plants, they are Beggiatoa bacteria, Topoca bacteria, they are filamentous bacteria. And one interesting thing about those bacteria is that the sulfur that they need is found, they have a conundrum. The sulfur that they need is found in the sediments, in the ocean floor. and while the nitrate is in the water column, so they have to navigate up and down, glide up and down to get those different nutrients. And we think that is a precursor, that's the forerunner of heliotropism and plant root formation, whereby plants shoot up into the atmosphere to get sunlight, to get CO2, and while they extend their roots deep into the ground to get water and other nutrients. But the lion's share of biodiversity comes from retroviruses, from what they call quasi-species. And the quasi-species stem from the extremely high mutation rates of those viruses, of RNA viruses. And basically, if we compare DNA and RNA viruses, the RNA viruses have a very much higher rate of mutation. Let's say regular RNA viruses on the bottom, they make one mutation for about each 1,000 nucleoside they copied, while for DNA viruses, we have about one mistake for about 10 million, for each 10 million nucleoside they copied. So as a result of the low fidelity of the RNA viruses, they found after several random replications, they would lead to formation of clouds of mutants they call quasi-species. Here we have an example of a quasi-species, a simple quasi-species here from the wild type of the virus in one would multiply, which is replication would form different variants that will form the quasi-species. And here the wild type of the virus here on the left side, like a round circle, after several random replications will lead to the quasi-species on the right. So here we have a quasi-species of retrovirus, the HIV-1, that was made from samples collected from several clinical sites in Cameroon, in Africa. And here for RNA virus, the SARS-CoV virus, this quasi-species here on the right. And then, so those quasi-species, the different strains of the quasi-species, that's what's going to combine with the bacteria here, CO number 3, to form the lineage that we have here. So, and in the tree of life, there is a tree of life, but the tree of life is basically of RNA genome from those viruses. They would form those different lineage that would evolve from one configuration to the next. And those configuration, that's what we call species. They are temporary, they are ephemeral. The species that the configuration actually they don't extinct, they don't go extinct. They just change configuration from one to the next with time. Evidence for the seed first theory, we will find evidence in the Cambrian explosion, and where if we start with the Cambrian explosion, which is the first appearance of animal with hard shells from carbonate or calcium shells and the arthropods, and then easily fossilizable, that we can find those the fossils in the record easier than the Precambrian animals, which were basically soft-bodied animals, less likely to form fossils, but they form fossil impressions on the sandstone. And we can see here, we can see also the shapes, the morphotypes or the phenotypes could look quite similar. And but if we go deeper in the deeper Precambrian rocks, like in the Dushanto Formation in southern China, we'll find those embryo fossils, embryo-like fossils, which are here at the different stage of development, which give a pattern at the deeper Precambrian rocks at the bottom here. After the global glaciation, we find those embryo-like fossils. And at some later time in the Cambrian, we'll have the soft-bodied animals. And in the Cambrian explosion, we'll have start having some hard shell animals easily fossilizable, which is about the same kind of pattern of evolution. If you take development from, let's say, from a mammal, from a vertebrate, we have the zygote here at the first stage to the germinal stage to the embryo. If you do an X-ray or a scanner, you cannot see it. But if you wait until the stage of fetal stage, they'll be visible on a scanner and an X-ray. Like if you take the CAT scan of this 30-year-old woman, which is like about 1 to 2 weeks pregnant, there's no indication in that scanner that you will know she's pregnant unless you have privileged information that she is. But if you wait 5 months later, when we take a scanner, you'll find in a uterus the lineaments of a vertebrate fetus developing. So, which is the same thing for the Cambrian explosion. We had to wait for the history of life. We had to wait for the Cambrian explosion to see those hard shell fossils easily fossilizable. But all the while, we had viruses and bacteria way back in time that at some point fused to form those embryo fossils I showed earlier, and then at later on started to form soft-bodied animals we've seen in the Precambrians. And also those animals in the first on the Precambrian area, they look about the same on the left side here. They look like leaves planted on the ground. They are not quite moving, so about similar type of phenotypes. But if we have to go to the Cambrian explosion, we'll have a certain diversification of shapes or forms or phenotypes. Which is remind us of the Ernst embryos. He was accused of forging, of faking his embryos, but he showed that the embryos of vertebrates, they look at the initial stage look quite similar, but they change. They start to diversify in phenotypes as time goes on. But we have photographs here of the same idea. Here at the first stage, at the first row, we have embryos of fish, amphibian, reptile, bird, and human, which look about similar. Looks similar, but when we start the next stage, particularly for the fish and amphibian embryos, they start to look different. So the viral evidence, if we look into the genome of animal eukaryotes, we'll find some the evidence as far as the viral presence in those genome. The Human Genome Project, sequencing of the Human Genome Project that was about 20 years ago, showed that a large fraction of most eukaryotic genome, they derive from retrovirus element. There's a lot of viral elements in the genome, and particularly in human, it's about 40%, 41 of almost 42% of the genome are formed by retrotransposons, which are basically viral elements. And if we add the 25 and 26% of the introns at the bottom of the pie chart here, those introns are derived from virus and viroids that give you more than 60% of components of the genome from viruses. And then the mammalian sperm cells with their craniocaudal axis resemble the caudovirals, which have about the same origin. It's a head-tail morphology, which is similar for both of them. Here, different morphology of sperm from different animals and human sperm cells as well. And here we have segmentation of the caudoviral tail, which is about similar to the vertebral column of a vertebrate. And then according to our theory as well, the Hox genes that are responsible for the segment anterior-posterior patterning of bacterians originate from retroviral genes as well, from retroviruses as well. And the sensitive protein, that the protein involved in the formation of the placenta in mammals, the coding sequences are made from envelope gene of human endogenic retroviruses. And then those sensitive protein are widespread found in all mammals. There are different morphotypes, different isotype of this protein for different mammals. And the bacterial evidence here, the bacteria basically show similarity with the eggs of mammals and eggs of oviparous animals. So Margita namibiensis descriptively has a lot of similarities with those with the eggs of animals about the size, about more than 100 times the size of bacteria. They're all spherical, they're visible to the naked eye, they're non-motile, and they have a large cytoplasm. And the Margita namibiensis as well, found in a low environmental oxygen, and also use nitrogen sulfur for energy, and they produce as product of metabolism ammonium. And for mammalian oocyte as well, mammalian eggs, they found in low environmental oxygen in the oviduct, and they produce nitric oxide and hydrogen sulfide. They use that for energy. And then for oviparous animal eggs, they need sulfur-rich amino acids for egg production. If you increase the sulfur-rich amino acid in the diet, they increase the egg production. And also if you look in culture media for human and mouse embryos, there is ammonium generated as well as it's the same thing also for the eggs of oviparous animals. They excrete ammonia like the Margita namibiensis bacteria. And the seed first theory also explained the origin of sex from the initial interaction of virus and bacteria. We have the later interaction of sperm and eggs, and that lead to sex. In conclusion, the early history of eukaryotes displayed the developmental pattern of fertilized vertebrate eggs consistent with the seed first origin of eukaryotic lineages. This theory and much more is published in my book that was published earlier this year, Ways of Nature. Thank you.
All right, thank you for this presentation. At this point, We have about a minute left for questions. So if anybody would like to be acknowledged by the author to ask a question, please use the raise hand button at the bottom of your screen, and they will acknowledge you to ask your question. If you're feeling shy, you're also welcome to type your question in the chat. I see that our next speaker is here. Daniel, you are welcome to put up your slides in preparation for your talk starting in about a minute. All right, well, that is about all the time we have for questions. So we're going to have to move on to our next talk. It looks like the speaker here ready to go. Perfect. All right. So without further ado, Daniel Schoenberg on the macroevolution of cell, nucleus, and genome size in odonates. Let's see. Daniel, we are not hearing you. Okay, I see you, but I don't hear you. Okay. I see, but don't hear you. Here, let me try muting and unmuting you. Unfortunately not, no. Hey, Daniel, I see you've rejoined us. If you want to give it another shot, we can see if we can make this work. I'm aware we're starting to press up against time. So if this isn't, if we give it a shot and it doesn't work again, it's fine. We can reschedule you into another session. So there's no need to panic about, what's going to happen with your talk. So let's give it one more shot. And if this doesn't work out, we'll take a brief recess before our final talk of the session. Okay, it looks like our speaker is still having some issues with their audio. So we are going to take a short recess until our final talk at 4:30 Eastern time, tapping internal states as predictive cues to maximize population growth in varying environments by Bing-Kan Xu from the University of Florida. So make sure you are back here for 9 minutes from now, and we will continue our session then. Internal predictive cues to maximize population growth in varying environments. Take it away.
Thank you for having me. Can you hear me?
Yes, we can.
Okay, great. So my name is Bing-Kan Xu. I'm from the University of Florida. I'm actually in the physics department. So this is my first time to be at the evolution meeting, and I'm quite delighted to be here. What I'm going to present today is work done by my student Leo Law, and it has been published recently. So to give you some context of our work, I've been quite fascinated by the phenomenon of bad hedging in biology. This is the idea that the population can diversify into multiple phenotypes as a way to cope with environmental variation. So, for example, in a bacterial population, some cells can switch into a dormant state, and sometimes called persister cells, that are tolerant to antibiotics. And by having these persister cells, the population of bacterial cells can survive antibiotic treatment. And similarly, in annual plants, people know that not all seeds germinate in the same year. Some of them stay dormant underground. And this is thought to prevent the whole population to be wiped out if a particular year is disastrous. Similarly, in insects, during development, some individuals can enter a diapause state, which is like a dormant state, and this allows them to survive some harsh environmental conditions. So the general idea of bad hedging has been verified. For example, in annual plants, you can see this negative correlation between the germination fraction and the variability in the environment. And this sort of qualitatively verifies the idea that when there is more uncertainty in the environment, you would like to lower your investment in certain phenotypes. However, when you try to verify this idea more quantitatively, these authors try to compare the predicted germination fraction from their model to the observed germination fraction, and you see that the fit is not great. They were able to realize that the fit can be much better when they included the dependence of germination on the seed density. And so, of course, we understand that germination is not a random event. And in fact, it depends on many environmental factors. For example, the temperature of the soil or the moisture in the air. And it is well known in the literature that these environmental cues are useful for predicting the environment if they are correlated with the environment. But to motivate what we want to study, we know that these external cues can be quite noisy, and also having to sense these cues can be costly because you need to develop organs to do that. And so we wondered if there are alternative sources of information that can help us predict the environment. So instead of looking outside for external signals, we would like to look inside the organisms. Of course, organisms are very complex systems that has a large number of internal states. For example, you can have metabolic states like the reserve level, or physiological or psychological states like the circadian clock. And we wonder if these internal states can be tapped as internal cues. Now, to qualify as a cue, we would require that these states are not sensors per se. For example, if you want to measure temperature, you will need some signaling pathway which acts like an internal thermometer. But I would not call that an internal cue because that's basically measuring some external signal. But for something like the reserve level, you're not hungry because you want to measure the abundance of the environment, food in the environment. So these states are not sensors per se, but as we want to study, maybe they can become correlated with the environment. And if so, they can be co-opted as cues for predicting the environment. So in the context of seed germination, you can think of seed age or size or the thickness of the husk as internal cues. And in this talk, I will focus on seed age as an example to illustrate this idea of internal cues. So to set up the model, we will follow what Cohen did in his classic work from the 60s. We are thinking about annual plants. Every year, the weather can be bad or good. In a good year, the seeds will germinate and they will have a high yield of new seeds. Whereas in a bad year, the yield will be low, approximately 0 maybe. And then the seeds that go dormant underground will have a probability of surviving until the next year. Now, if you simulate this process, you will see that the population size will fluctuate over time. But in the long term, there is a trend line. And you can use this slope to represent the long-term growth rate of the population, which is also equivalent to the so-called geometric mean fitness of the population. If you do a little bit of math, you will have this expression for the long-term growth rate, and you can see that it depends on the germination fraction. This function has a maximum when G is approximately equal to P, which means that to maximize the population growth, the germination fraction should be roughly proportional to the environment probability. And this has been used as a rule of thumb in studying bed hedging. And people have also considered the presence of environmental cues. So in this case, each year you will receive a cue like the temperature or seed density. And depending on the cue, the germination fraction can change. And so if the population utilizes this environmental cue, you can see that the population can grow faster. And it is known that this is useful only when there is correlation between the environment and the cue, which is represented here by this conditional probability of the environment given the cue. The fact that this is not a constant shows that there is correlation. So in this simulation, I set the cue to be 70% accurate. And with this, we can also calculate the optimal germination fraction. And as you can see, this fraction can depend on the cue. So move on to what we want to study. Instead of considering an external cue, we would like to study an internal cue, such as the seed age. So this is represented by this diagram on the right, where the states S represents the age of the seed, and the blue arrows represent going dormant, so the age is increased by 1. And then the orange arrows represent germination, so that a new seed has age 0. These parameters on the arrows represent the probabilities. So will this be useful? We know that in order for the Q to be useful, there has to be temporal correlation. In this case, the seed age is like a memory of the system. So in order for that to be useful, the environment has to be temporally correlated. So for that, we simulated the environment in this particular way, so that if you think about the epoch of consecutive good years or bad years, the duration of the good years is geometrically distributed, which represents like a Poisson process that doesn't have memory. So disasters is like a random event, whereas the duration of the bad years has a narrow distribution, which sort of represents that the disasters typically last for a certain number of years. So with this setup, we can study now whether seed age is useful as an internal Q. So here is our results. First, we verify that indeed the seed age becomes correlated with the environment. Here I'm again showing the conditional probability of the environment given the seed age. The fact that this is not a constant shows that there is correlation between the environment and the seed age. Now, I want to emphasize that this is, the seed age does not have to be correlated with the environment a priori. And what you see here is a result of selection, so that the distribution of ages in the population is biased through the selection process that gives rise to the correlation. Now, with this correlation, you can expect that the optimal germination fraction can now depend on the seed age. And that's indeed what we see, as shown here by the blue bars. And we, as a cross-check, we simulated a case where the environment is not temporally correlated. And in that case, the germination fraction stops being dependent on the seed age. So that verifies our predictions. And in fact, I would like to mention that there's a previous study which argued that there is no benefit for the germination to depend on the seed age. And we realized that in that study, they considered uncorrelated environments. So another thing that's interesting coming from this study is that we can now think of the internal states as a capacity for the memory of the system. So in this transition diagram, we can cap it off at a certain number of states. This is like to say, after a certain age, I stop counting my birthdays because I'm just old. And so it's interesting because now we can vary this number of states to check the memory capacity. In fact, some of these parameters will correspond to special cases. When L equals 1, this reduces to the simple bad hedging case that doesn't have memory. Whereas for L equals 2, this is equivalent to our Markov model, which is like phenotypic switching. And as you can see, when the capacity of the memory increases, the population can grow faster. You may ask, can we tell experimentally whether there is a memory capacity in the system? And in fact, it is possible by studying the distribution of the duration of dormancy. And I will come back to this in just a second. Okay, so there are a few aspects to discuss from the results. First of all, our model predicted that under this particular environmental temporal structure, we will see this dependence of germination on the seed age. Now, we tried to find experimental results on this, and the best we could find is some experiment done by Philippi from the late 80s. And he studied several desert annual species, and he verified that the germination fraction indeed changes from the first year to the second year. Now, year 1 actually corresponds to age 0 in our model. And so in order to see this more detailed structure of age dependence, you actually have to go beyond year 2, and we couldn't find such experiments. Now going back to diapause of insects. So in this particular model, the authors consider the case where you go into diapause, and when you come back to this branch point, you have the same probability of entering diapause again. So this actually corresponds to our model with one state. which is memoryless. And people have studied other scenarios. For example, in this work by Tauja Perka and Eistock, they modeled the diapause such that you can enter one round of diapause and then you have to exit. And this is actually a special case of our model with two states, where you have to exit after first period of diapause. And finally, when I come back to microbial phenotypic switching, this is probably the best studied system quantitatively. And in this example of Bacillus, the cells can switch between a motile state and matrix producer. And these authors studied, under microfluidic devices, the switch between these two states. So when they produce extracellular matrix, the cells can stick together to form these long chains. And they can they can record the video for a very long time and measure the statistics of the duration of these states. They found that the motile state is distributed exponentially, whereas the chaining state has a very narrow distribution. Now, this is curiously reminiscent of what we found about the duration of the germination and the dormancy periods. So as I mentioned, this can be used as a signature to test the existence of memory. And I hope that such experiments can be done in seed age as well. Okay, to conclude, we found that internal states of organisms can be tapped as cues for predicting the environment. And also these internal states represent a memory that can be tested in experiments. There's also interesting connections to information theory, which I don't have time to go into here, but you're welcome to check our publication. And I thank you for your attention.
Excellent. Thank you for that talk. So we have about a minute before our session ends. And so you are now free to answer questions. It looks like we've already got one from Albert.
Hello.
Hi.
Can you hear me? Hi. Thank you for the talk. I think this is very interesting. I'm just curious about the age-dependent structure that you set up when for the internal state of the seed. So like you have these different rates of moving across different age groups. So how are these rate parameters being formulated? Is it always the case you have a higher transition rate when you grow older, or is it like the other way around, or is it like no specific?
No, so in our model, we basically assume that the germination fraction can depend on the seed age, but we maximize the population growth rate to find the optimal dependence, and this is what you get. So
yeah,
we don't assume, yes.
Mm-hmm. Right. So like these G's that you have on the other slide, where you have these sort of like a Markov chains, like they don't have like a sort of like a constraints in what values.
No,
Okay.
exactly. When we do the optimization, we don't constrain it. But as I mentioned, if you constrain the number of states, then the germination probability will have more structure.
Okay. Right.
So for example, if you only have 2 states, then it is constrained to be Markovian.
Yeah.
Like you said.
Thanks.
Excellent. Well, this concludes our session for the macroevolution session this afternoon. Let's see, after this session, it is a short break from 5 to 6 Eastern Daylight Time, at which point we'll resume with our concurrent sessions. So thank you to all of our speakers, and thank you to all of you for attending and participating today.
Thank you.